Welcome, everyone. Before we begin today's webinar, there are a few topics of housekeeping I would like to go over. First, all questions can be put into the Q&A chat. We will answer all questions after the presentations. And second, we will be sending the full recording to all registrants later this week. And now, Evan, if you would like to begin.
Yes. Can you hear me okay?
Yes.
Awesome. Well, thank you for that introduction, Emma. I'm very happy to introduce the speakers we have for today. Be myself, Anthony Eritano from SCIENION, and Seyoum Ayuhenie from MatTek. I'll be talking about some phenotypic assays that we've developed and systems we've developed for cancer research. Then Anthony will talk about the spheroONE, which is an automated dispense system for improving the workflow for using 3D models, tumoroids, etc. And then last, Seyoum will talk about work he's been doing at MatTek in developing primary colorectal cancer tumoroids and using this new workflow that we've developed to really show how you can automate and improve the data collection and quality of data that you can get, as well as show some comparisons between the primary colorectal tumoroids and immortalized cell lines, which we find very exciting. Now, let me start the story. Is why 3D cell models? And if you look at the history of drug discovery, drug development, it's really been based on initially on patients, using the patients as the test tube, if you will. And you can get all the effects of there, but what you really want to do is move from the patient to something that is higher throughput, if you will, something as less costly. And traditionally it's been biochemical or cell-based assays. But the reason 3D becomes important because you can recapitulate the in vivo environment, all the genetic changes, all the gene expressions, the protein expressions, etc. And from those get data that you can then mine through and really push the boundaries of personalized medicine, personalized therapies. So I mentioned, 2D cultures have been used for decades. In fact, HeLa cells came out in the 1950s, and those are thought to revolutionize. But people quickly found the limits of 2D cell cultures. Plastic is not natural. 2D dimensions is not natural. So if you can go to something that it better represents the organs and the tumors inside the body, you can get better data, better productivity. And there's been a huge increase in the past probably 10, 15 years in organoid area, in ways to do 3D cultures, and using patient-derived cell lines, etc. And that has really revolutionized the whole drug discovery industry. Some of the models that we'll be talking about today are spheroid models. You can also do spheroid co-cultures, which better recapitulate, especially for tumor models. And then doing actually tumoroids, and this is something that MatTek has actually been a leader in for the past 20-25 years, is tissue models for tissue on chip, tumor on chip, etc. And so Seyoum will talk a lot about that, a very exciting area. But I think what we really want to emphasize here is the development of 3D models is really at a crossroads. It's now being used extensively in pharma, but there's still not the tool set that you need to move this into that next phase of drug discovery, the higher throughput, where it becomes very reliable and very reproducible. And that's really where we're focused on, using our Pu.Ma system to automate assay steps with 3D cell models. The spheroONE allows you then to manipulate and dispense and disperse, identify the 3D samples you want to run in your assays, and place them in the consumable, the plate that you need for your assays. And then ultimately, MatTek using these as a tool, running viability assays, looking at response of therapeutics, using these patient-derived, these ex vivo models, which give you better data and better predictivity for drug development. So let me start with our Pu.Ma system family. This is a series of microfluidic flowchips. with a benchtop system that uses pneumatic pressures to move fluids through microfluidic channels. It allows you to do fluid exchanges without disturbing or perturbing your 3D cell sample. It's also compatible with a wide variety of downstream data collection, including imaging, supernatant sampling, and phenotypic profiling of 3D cell models. Some of the features that we like to highlight is really this protected sample chamber. And this is unique to our flow chip, which allows you to, again, manipulate and do fluid exchanges with your 3D cell model without disturbing it or moving it or damaging it. Because we're a microprocess controlled system that you can schedule your media exchanges, you can schedule supernatant sampling, you can do that automatically and look for dynamics in changes of metabolism, immune response, other biological factors that are important, phenotypic changes that are important. And at the end, you can do immunofluorescence staining, which is a very complicated multi-step process. And we allow you to do that in a much higher efficiency. And again, be able to grab images that are really beautiful images of your organoid models. Now, the flow chip is a injection molded plastic piece that's sealed on the bottom. So it's a very simple device. Essentially, each lane in the flow chip is a sample run, and the sample well is connected to reagent wells through microfluidic channels on the bottom. Look at the cross section. It's about a 20 microliter volume, and this protected sample chamber is about a 1 millimeter diameter with a very thin, optically clear plastic on the bottom. Again, perfect for doing high resolution imaging. You also get about a 95% fluid exchange, and so there's about a 1 microliter residual volume in your sample chamber. So when you're doing sample exchanges, reagent exchanges, you get 95% exchange without drying out or disturbing your sample model. So how does that work? This video shows you how the fluid exchanges occur. When the sample is in the protected sample chamber, we use pneumatic pressures to either push fluid out through these side ports or push fluid in through the side port. And so you again can do that exchange, and there's 8 wells connected, so you can do up to 8 different exchanges through this sample well, and allows you a lot of flexibility in your assay steps. Let me mention briefly a couple of new systems we have out. We have our Pu.Ma system EC, environmental control. So on your benchtop, you can now control temperature, CO2, humidity. You could also take our regular Pu.Ma system, put it in an incubator. But if you don't want to tie up an incubator or you want to have more flexibility, Pu.Ma system EC is perfect for you for that. And we've also come out with low attachment flow chips. So now you can form 3D models in your flow chips. You can also do co-cultures. I've done some exciting work where you had immune cells into them, and the immune cells can then move around, find your tumor models, infiltrate, kill the cells, and you can monitor that whole process over multi-day periods. So that all gives you this complete workflow from cells to data, where you can either form your organoid in the Pu.Ma flow chips or load them in. You can then do incubations of compounds, media exchanges, supernatant collection. And at the end, you can then do automated immunofluorescence staining and imaging, do complete phenotypic analysis of response of your tumor models to samples and perturbations. So let me give you a quick example of that. This is work we published in SLAS Discovery back in 2022, showing what we call multifunctional profiling. So again, with your sample in the flow chip, you can do a viability assessment. You can look at suite of factors. At the end of that, you can then fix it in the flow chip, do immunofluorescence staining for biomarkers. And I'll just show you today briefly the viability assessment part. So in this workflow, we are using agar roll plates, which have microcavities where you can form thousands of small tumoroids in the plates. You can then collect those, and it's about a 2 to 3 day process to form nice firm organoids or tumoroids in these plates. So after, say, 3 days, you collect those, transfer them into flow chips. In this case, we're doing about 1 to 5 per well because we're doing manual transfer. and you want to make sure that you filled up all the wells and you got them nicely dispersed. And then once in the flow chips, then we use them in our Pu.Ma system to do compound additions, to do incubation, and ultimately to do viability staining. So the setup on the flow chip, there's 32 samples. So in this case, we did 4 repeats, and over one flow chip, you could do 1 to 3 steroid dilution, get 8 concentrations. If you want to do 2 repeats, then you could do either 2 compounds at 8 concentrations, or you could do a higher number of concentrations for the compound. So it's a very flexible setup. Again, compatible with 384 well liquid handling, compatible 384 well imaging systems. So once the concentration's been set up, we use, again, the Pu.Ma system to do the incubations, and then we did imaging at the end with the automated microscopes. Here we're showing work we did. We looked at doxorubicin and oxaliplatin, and you can see a nice concentration response. Phenotypic changes from live, which is green. We're using cyquant green as a live viable cell stain, and ethidium homodimer as a dead cell stain. You see the very nice transition in there. And these images were acquired. These are confocal maximum projection images acquired from the CQ1. Now for image analysis, we wanted to really kind of push the boundaries and use AI-assisted algorithms to do this. So we've developed, these are open source algorithms, and we can give more information if you're interested on this, but really take the images of your spheroids, train the AI recognition and thresholding algorithms to identify what's a spheroid. And we do this in the green and the red images separately. That data set, then the area, intensities, total intensity area, all the sort of usual suspects of information you would get out from these, we could then use those to look at viability and determine IC50. And so for the images you show for that data sets, here are the concentration response curves for doxorubicin, oxaliplatin. We saw very nice IC50 curves and slopes, and actually really good agreement with literature values for HCT116 cells. So we're very excited about this, and I think comparing this then to the primary colorectal cancer organoids or tumoroids that Seyoum will be talking about later, shows some very interesting results. So in summary, I think we brought the Pu.Ma system to market to really help researchers who are using 3D cell models and struggling with manipulation running assays with them. So we improved the efficiency, we improved the capability that you can do with those on a benchtop automation system, allow you to get multi-parametric data. And we're pushing the analysis, phenotypic analysis, using AI-assisted data analysis, which I think is really the future for drug discovery. And design the system to be compatible with automated systems, whether it be liquid handling systems, be spheroid dispense systems, be imaging systems. And I'll now transfer it to Anthony, who will talk about doing automated spheroid dispense with his spheroONE system.
Hi, everyone. Thank you for attending this webinar, and thank you to Evan for that introduction. And today I'm going to talk about the spheroONE, which is a machine that can isolate individual spheroids. So next slide, please. So the spheroONE, it is basically a game changer in the field of doing drug screenings, toxicity assays, and 3D model applications. And its main applications are for sample preparation for drug testing and organoid culture and manipulation. And what the spheroONE can provide is a standardization of 3D models, selection of functional 3D models, and enhanced assay qualities and reproducibility. Next slide. So most of us here have worked with spheroids before, and we can know that they can be very delicate, a very delicate system to work with. And that's because these 3D models or these cell aggregates, tumoroids, organoids, they're very fragile, and they're very, very hard to manipulate without causing damage. And also imaging can be very, very tricky, especially when you have two spheroids that are too close together or they're overlapping. So you really get there's really a hard time isolating what is part of one spheroid or the other. And current workflows using ultra-low attachment plates have a very low throughput. It takes time, and working with them is complicated. And the other downside to using these low attachment plates is that you actually see a huge degree of heterogeneity in terms of size, biomass, and actual functionality. And because of all this heterogeneity, it reduces assay reproducibility and can make interpretation of your data very difficult. So the solution is to use the spheroONE to basically automate, sort, and isolate spheroids, organoids, or tumoroids. Next slide. And what makes the spheroONE so powerful is that it uses a precise nano to microliter dispensing technology. So we have a nano dispensing capillary, call it an NDC, and this allows us to isolate spheroids that are between 100 and 600 microns in diameter. But it can also dispense liquid, reagents, samples in solution. So it's very versatile when it comes to what it can actually dispense. And what's really important is that it's very gentle when it comes to handling these spheroids. So we have a electromagnetic microvalve that makes sure that the drop that is coming out of this capillary is done very delicately, so the spheroid itself is not damaged. And this instrument can also give us a highly reproducible drop volume. So the drop volume is around 500 nanoliters per drop, and it's very, very consistent in that. And the other cool thing about this machine is that it's compatible with any substrate you can think of: 96 well, 384, 1536, slides, or any kind of custom substrate that you have, like a biochip or anything like that. And the dispensing process itself is highly precise, and we can actually deposit spheroids in a correct position on the micron scale. Next slide. And the thing that makes this machine really powerful is it uses image-based particle detection for its isolation. So what we can see is that if we look at the NDC, it's divided into 3 zones. You have the bubble zone, which represents the volume of liquid that was dispensed previously in the previous drop. You have the ejection zone here there in green, and that is basically the volume of water that's going to be dispensed next in the next drop. And then you have the blue zone, which is the sedimentation zone, and that's the zone where the spheroids tend to fall down. I consider it sort of a safety zone, because if there's a spheroid in there, there's a chance that it can get dispensed along with the spheroid in question, so you can end up with two spheroids instead of one. But the other concern with using spheroids is that, they're very hard to make and their numbers are minuscule, so you really don't want to waste your sample. And what's really nice with the imaging software of the spheroONE, it can allow you to recover your sample. So let's say there are 2 cell masses inside that ejection zone. Well, the machine is not going to isolate them because it's not a single spheroid, but what it will do is it will dispense it into a recovery tube. And the same thing if there's a cell in the ejection zone and a cell in the sedimentation zone. It's not going to just toss them, it's going to put them into a recovery tube. And you can reload that recovery tube into the system and run the samples again. And this is all made possible due to the unique type of illumination that the machine uses. So the spheroONE uses dark field illumination. So this captures all the scattered light, and it eliminates the need to do any kind of staining or vital dye staining to visualize these samples. And every image that is taken is captured and saved automatically for your records. Next slide. So this is the spheroONE in action. I want to state that this video, it looks slow because it's going at a quarter speed because of bandwidth issues. But just know that that clock actually represents the time it takes. And as we can see, if we imagine this going 4 times faster, that the spheroONE has a fast sorting time. So we can get the machine to isolate 20 spheroids a minute. It has high efficacy of isolation. So we can obtain basically up to 100% confirmed isolations in a specific spot. And it's also highly customizable when it comes to isolation. We can gate samples based on size and shape. And if we look at some data from our colleagues in Lyon, we can see that when we have a very broad gating system, we're collecting 100 to 500 microns, we can see an array of sizes of spheroids that were isolated. But then we can gate the size to between 350 and 500, and we can see a uniform sample of spheroids isolated. And same thing when we look at the lower end between 200 and 250 microns. Next slide. So to go over the workflow again real quick for the SPHEROONE part, first we grew HCC 116 cells colorectal in a microcavity plate to do bulk spheroid production. Those were then transferred into the sample reservoir and loaded into the SPHEROONE with the 3D flow chip plate. And then during this process, the SPHEROONE takes images and it checks criteria. So the first criteria is the object that detected the spheroid cell aggregate the correct size? Is it in that size range we specify? Does it match the elongation range? So the elongation range gives us shape. And is it a single spheroid? So if that particular spheroid passes all of that criteria, it'll be dispensed into the well. Next slide. And so this is just some sample images from one of the flow chips. So if you look on the left, you'll see the pre-isolation image. So the machine takes images before you dispense, and you can see there's nothing inside of those wells. And then it goes in through the isolation process where it isolates individual cell aggregates of whatever size you want. And then it can take a post-isolation to show you where that spheroid was placed. And if we zoom in and we look inside the well, we can actually see that each well has an isolated spheroid in it. Next slide. And the really nice thing about the SPHEROONE is that it's, when compared to manual isolation using a pipetter, we can see a higher level of viability and morphological identity. So with the SPHEROONE, we can see that the SPHEROONE dispense, we have a nice, almost completely circular spheroid isolated versus manual isolation. And in this case, we can see that there are two spheroids that were manually isolated. And this is because you tend to hedge your bets when working, when you're manually pipetting. You rather have two spheroids in one rather than having a well of empty. So it's a way of hedging your bet to get the numbers you need. But with the SPHEROONE, you don't even need that. So the SPHEROONE compared to manual shows that 97% viability compared to manual. And it also shows that for these experiments, we have 98% spotting efficacy, which means where we want that spheroid to go, it got there 98% of the time. So with the SPHEROONE, we can isolate spheroids or organoids that are more uniform in shape. We can show that the spheroids have a high level of viability to confirm how gentle the machine is with the machines, and it can be isolated into a position where we want it to be. And next slide. And with that, I would like to turn the presentation over to my colleague Seyoum, who's going to talk about the colorectal cancer tumors.
Seyoum?
Yes. Thank you. Thank you. And next slide, please. Yeah, before I talk why we need primary, patient-derived primary tumoroids, I have to discuss a couple of things showing the problem, what the existing testing methods are contributing. So drug development is a very expensive and time-consuming process. It takes about 12 years and $2 billion of expenses to get one successful drug by the pharma company. And then as a result, drugs are extremely expensive. Cell lines have been used for a long time, but they lack structural features of organs, and they don't predict human responses. And as to the preclinical models, even though they have been used to address some of issues like metabolism and so on, they are poor predictors of drug toxicity and efficacy. And about 90% of the drugs that passes the preclinical testing fail during clinical trials. So this, particularly for oncology drugs, about 75% of the drugs fail during clinical trials. So the current system has drawbacks and shortcomings, and is not predicting the human responses. So we move to the patient-derived in vitro cancer models, because they are taken out from the patient and they are more physiological, and they can predict treatment responses in humans, and they can also identify new genetic markers of drug resistance. Due to this, they have high prediction capacity of human responses. Next slide, please. And the technology that we adopt to get tumoroids, large-scale tumoroids, has two steps. In step one, we take samples from a patient tumor, and then those are 4-millimeter biopsies, equivalent 4-millimeter biopsies. Then we chop them up and isolate tumoroids from the patient, and we grow them in vitro. Then we expand them by passaging them a couple of times, so that they don't have to change their phenotype. So we are talking about low passage tumoroids. And then once we expand these cells, we just create a biobank, and that biobank will help us to generate more tumoroids. And from there, from our biobank, we can take a vial and put them to high performing plates with microwells. In this case, for example, we use a 12 or a 6-well plate. The 12-well plate has microwells that you can generate up to by taking one vial of cells and then disintegrate them and dispersing them into these wells. You can generate them within about 13 days time. You can get up to 10,000 organoids from one vial of cells. Or if you use a 6-well plate, we can generate up to 20,000 organoids. So what it means is, okay, if we have 20,000 from one vial, then we have millions of organoids that we can work with from one 4-millimeter biopsy punch of the tumor. So which is a huge endeavor in this aspect. But the next challenge that we are facing now is, okay, we generate this numerous tumoroids, and how do we put them onto plates, 96-well plates, 384-well plates for high throughput drug screening? That is the challenge that we are facing, and that's where these new technologies are going to help us, like spheroONE or Pu.Ma. So how we can do it is, if we are going to do it manually, the cells are going to be, it will take us about probably to make five kits of 96-well plates. Probably we have to spend the whole day. But if we harvest them using spheroONE, probably in 5 hours we can load up to 50 96-well plates. That's the power of these equipments, and we need those helps, and that's how we try to integrate this system to the tumoroids. Then we can put 96 well plates, or from one plate of organoids of the 6 well plate, we can make up to 200 96 well plates. We can load 209 or about 50 384 well plates, and that is powerful for high throughput screening, what the pharma industry is looking for. Then you can do staining, and you can image them, and you can analyze them, the image, using AI technology. So those are the things that we have to entertain in the coming years. Next slide, please. So I will give you some few examples how this work is going to be. This coordination is applicable, or integration is applicable for cancer research to screen oncology drugs. So we have primary colorectal cancer cells that we generate, and we compare them with the known cell line, colorectal cancer cell line, and then we try to see some of the effects that we are seeing as the differences. So if you look at here, Dr. Robinson, that drug doesn't have much difference between the cell line and the primaries. So next slide, please. Over here, if you look at this one, then you can see few differences, and the concentration are not the same. But if you look at at 0.8 micromolar concentration, the primary cells are sensitive for this particular drug. But you look at the cell lines, they are more resistant at 11.1 micromolar. So that is almost about 10-fold lower sensitivity in the primary cells than the cell lines. So whatever you are predicting in the cell lines may change the human responses in terms of toxicity. Next slide, please. This is another example, which is a chemotherapeutic drug also. So if you look at here, at 200 micromolar concentration, the cell lines are fine. But with 5 micromolar concentrations, the primary cells start to show some kind of cell death. And that is intensified when you have 25 micromolar. So probably over here that you can see close to 20-fold difference in terms of concentration that you have to use to kill the primary cancer cell lines or primary cancer tumors. So whatever you are, here, for example, if you are estimating drug responses based on the cell line, the standard cell line, HCT116, you can take it over 200 micromolar, and you can model it for the human consumption, and then what it means is it could be more toxic to human, and the side effects could be higher. And what it means is there will be drug attrition by the patients. Next slide, please. And this is a targeted drug, cetuximab, which inhibits blocks the EGFR receptor, and as you can see it, the cell lines are fine with higher concentration, like 100 microgram per ml. But if we look at the primary tumors, you can see cell death starting from 68 microgram per ml, or 13.6 microgram per ml down. So you can also see here a difference in responses. So those are type of things that this sensitivity is it true, because we are now working with only tumoroids over here, but to make sure that this type of responses are happening in a microenvironment when the tumor is grown. I will show you one more example where we have basically started to make to incorporate these tumoroids into a 3D tissue model, which makes it more similar to the human microenvironment. Next slide, please. So what we did here was we took the cells, the tumoroids, and put them into a microenvironment where matrigel, where collagen and fibroblasts, endothelial cells, and dendritic cells are present. So we recreate the tumor microenvironment. And as you can see, the histological structure between the in vivo and the in vitro are very similar. And then we look for the drugs, dosing the drugs at different time points. And as you can see, cetuximab also shows by day 11, you can see a cell death at lower concentration, which is 0.25 microgram per ml concentration. That indicates that the primary cells, primary tumors can be more sensitive to these drugs, and they can be, you can predict correctly what the human response are going to be for this type of drugs. Next slide, please. And the conclusion: these 3D cancer models can be used as a preclinical tool for oncology drug screening, and the primary tumors show more sensitive to few drugs compared to cell lines based on the cell line based assays. And this is a good example of a bioconvergence where we show integration of large scale tumoral expansion in the lab, and then the tumoroid sorter and dispenser technology to put them into a plate, and then imaging capability using like a digital microscope, like a microscope, and image analyzer, and that leads to a complete workflow to advance the next generation of drug screening systems. With the systems, I think we will have a better chance of modeling, drug responses in a very short period of time. Thank you.
Thank you very much, Seyoum. I'd like to acknowledge the other people at PFI, Andrea Merlou, Emma Cromwell, Katya Nikolov, who participated, from SCIENION, Jerry and Barbara, the group at Cellenion in Lyon, who have really developed the spheroONE, and then the MatTek, Dylan and Matteo, who are participating in Seyoum's group, helping develop the tumoroids and patient-derived cell lines. And then one shout out for the MPS World Summit, which is occurring in a couple weeks. We'll have a couple posters there. One really going over a lot of the material we showed at this webinar. And so if you're there, please stop by. If you'd like a reprint of the poster, we can certainly send that to you. And then Seyoum has a poster detailing more of the colorectal cancer tissue models that they've been developing. So please, if you're at MPS Summit, stop by. And if you're not, we are more than happy to share this information with you. So with that, let's open up for questions and answers. Emma, would you like to?
Yes. So we have a couple of questions. First, this is for Anthony. What kind of media is compatible with the spheroONE?
So the media, the spheroONE itself, can actually dispense a wide variety of media. So the only real tricky part is that we like to keep the BSA level on the lower side, or maybe around 5%. But other than that, it can dispense any kind of cell culture media. We normally resuspend our spheroids in a 20% PEG solution because that helps with the sorting ability. So there's a good amount of viscous solution it can handle. It's just we don't want to overload it. But yeah, any kind of media is possible to be dispensed.
Cool. And then next one is for Seyoum. What is the advantage of using patient-derived tumoroids compared to existing cell lines or spheroids?
Yeah, basically the spheroids are a simple collection of cells or aggregates, and they are not complex. They lack polarity, but the tumoroids have polarity, and they are more complex, and the tumors are more closer to the human organ or the organ that they are coming from. So they represent exactly how the cells looks like in a human, within the human body. But the spheroids are just a simpler type of collection of cells that lacks polarity, and the predictions that you make based on these cell types, spheroids, may not be comparable to that of the tumoroids, and the tumoroids are more superior in terms of prediction of human responses.
Great. And then next one is for Evan. How many supernatant samples can you collect for each tumoroid sample?
So in each sample lane, there are 8 wells connected to your sample well. And because we have to use some wells for media exchange, etc., practical is there's 6 time points you could collect, 6 samples you collect per sample. And you can set that up over any time frame you want. So if you want to look for every 3 hours, 6 samples give you, say, a 1-day response. That's possible. If you wanted to look for a multi-day response, collect a sample every 12 hours or 24 hours, that's possible too. We also have the ability to reload wells, and so if you wanted to collect a series of samples and then reload that with media or with compounds and continue that experiment, that is also possible. So a lot of flexibility.
Great. And then next is for Anthony. Can the spheroONE isolate IPS organoids?
So yes, so between the work we do here in the U.S. and our collaborating staff in Lyon, we have isolated many different types of tumoroids and organoids. We've isolated mouse organoid, lung organoids, kidney ducts from mouse, pancreas duct from human, gut, and IPS-derived brain from mouse and human, in addition to human liver organoids. So we have done a wide array, and the list is growing. So yeah, so we can isolate pretty much any organoid as long as it fits the size limitation between 100 microns and 600 microns.
Fantastic. And then next is for Seyoum. Can you mention some of the endpoints that can be obtained from the AI-assisted analysis of drug-treated organoid images?
Yeah, as Evan mentioned it, they can, you can look for intensity, fluorescence intensity of, say, for example, if you are staining with variable dead staining, you can look for intensity marker, and you can graph it. And also based on that, you can also look at if you are looking getting to certain areas, probably the one Evan mentioned, like the IC50 determination based on the analysis. That is a great thing also, because you can do dose range finding using these tumoroids, and with AIS you can have a fast track to look for IC50 concentrations and so on. So it can be used for those type of things. And also, if you are going to, you can take it that concept. If you are if you stand for different markers, if you have different cell types in the tumoroid, if you have different cell markers, then you can quantify those cell markers, and you can see the location. You can see the intensity, whether those cells are dying or living, are alive. So you can make those type of comparisons, and I think that opens up a new area where we can have more of marker identification, particularly associated with surface markers identification using these technologies.
I would add to that. A lot of high content image analysis really been developed around 2D cell images, where you can identify each cell, you can segment that cell, you can look for membrane, you can look for the nucleus, you look for cytoplasm. So it's just amazing what you can do when you can identify your cell and then do analysis on that. Once you go to 3D, overlapping cells and everything kind of gets mushed together. So your 2D image algorithms do not work very well with 3D cell models. And that's where we feel AI-assisted algorithms are going to be really important to pull out the information that Seyoum was talking about, the information that drug discovery scientists need to develop that next generation of oncology therapeutics. Again, because of the complex nature of the 3D, using AI algorithms, deep learning algorithms, is I think going to be critical to that analysis in the future.
Great, thank you. And then the next question is for Anthony. Will fluorescence capability be available for the spheroONE?
So the fluorescence capability is actively being pursued. So they are definitely working on that. I don't know a time frame, but it is high on the priority list of things that we are looking to add to the spheroONE to improve its functionality.
Great. And then next question is for Evan. What imaging systems are compatible, or is the Pu.Ma system compatible with?
Well, the Pu.Ma system flowchip was designed with the 384 well format, and so it's conforms to the SLS standard for 384 well plates, and so any imaging system that can handle a 384 well format you can put our flowchips in there. Now the thin plastic bottom allows you to go up to 100x, oil immersion, water immersion lenses, and so it's compatible with really any high resolution imaging system that you have out there. With the caveat that you really can't do, for example, any somewhat super resolution is not really compatible with it, but most people doing high-con imaging are using automated imaging system using epifluorescence confocal imaging system, so it's completely compatible with all those. I will also mention that because of the design, you're able to locate your organoid sample very easily. It's located within a 1 millimeter target. It's located right at the bottom in that protected sample chamber. And so being able to find and grab that image is much easier in our system than if you were using, say, a 96 well plate or a matrigel dome or some of the other common ways people use to form and do assays with 3D cell models.
Great. And then we have one more question for Anthony. If anyone else has any more questions, please put them into the Q&A and we can get to those. So Anthony, can the spheroONE dispense multiple spheroids into a single well?
So that's actually something I wish I could have touched on a bit more. But the software itself can allow you to dispense 1, 2, 5, 10, 20, 30 spheroids in a single well. So the software is very flexible on how many spheroids you want per well. The only real limitation is how many of them can fit in your well. So any number of spheroids you want in a well, it can be done.
Great. So that is all the questions we had. If anyone else has any questions, please feel free to either put them in the chat. I will keep the webinar open for a few more minutes. Or alternatively, you can email Evan, Anthony, or Seyoum. Their emails are also in the chat. Otherwise, Thank you all for this wonderful webinar and presentations. And I'm excited to see where all of these instruments go and the new research that comes out with them.
Yeah, thank you much, Emma. Thank you, Anthony and Seyoum. Look forward to seeing everyone at MPS Summit.